In this video we will talk about single gene syndromes. So, single gene syndromes, also known as monogenic disorders. They are a group of genetic disorders that are caused by mutations or alterations in a single gene. These genetic mutations can be inherited from one or both parents. Or, they can occur spontaneously during development. Single gene syndrome include Rubinstein-Taybi, or broad thumb great toe syndrome, and Alagille syndrome, type 1. Let's start with Rubinstein-Taybi, or broad thumb great toe syndrome. Frequency of Rubinstein-Taybi, or broad thumb great toe syndrome is 1 out of 125,000. It is autosomal dominant disease. Genetics of Rubinstein-Taybi, or broad thumb great toe syndrome is del 16, p13.3. which means deletion on chromosome 16. At arm P, at band 13.3. Let's move into further details of Rubinstein-Taybi, or broad thumb great toe syndrome. Its etiology is that cyclic adenine monophosphate response element binding protein is a histone acetyltransferase that functions as a transcription factor for glioma-associated oncogene proteins concerned with patterning of skeletal elements. The deletion creates a haploinsufficiency that leads to this syndrome. Are you confused? Now, let's not jump directly into the etiology. Let's understand the normal pathway first, and then deletion that creates a haploinsufficiency that leads to this syndrome. This is exterior. This is cytosol. And this is nucleus. Many agents, including neurotransmitters and hormones, act to increase the intracellular level of cyclic adenine monophosphate. Typically, such a ligand binds to its cognate receptor, most often of the 7-transmembrane domain class of receptors. Ligand binding of the receptor leads to the activation of a coupled heterotrimeric G protein, whose activated subunits stimulate one or more of the adenylyl cyclase isoforms, the enzymes catalyzing cyclic adenine monophosphate production. Production of cyclic adenine monophosphate is counteracted by the action of phosphodiesterases, the enzymes that cleave cyclic adenine monophosphate. The cyclic adenine monophosphate-dependent protein kinase contains 2 regulatory subunits with binding sites for cyclic adenine monophosphate and 2 catalytic subunits. The tetrameric protein is enzymatically inactive because the regulatory subunits block the catalytic sites on the catalytic subunits. Binding of cyclic adenine monophosphate, which occurs cooperatively, causes dissociation of the catalytic subunits, which activates the protein kinase. When the cyclic adenine monophosphate-dependent protein kinases are activated by cyclic adenine monophosphate, they translocate to the nucleus, where they phosphorylate and activate cyclic adenine monophosphate response element binding protein. This stimulates transcription of genes controlled by the DNA regulatory element, cis regulatory element, which is regulated by cyclic adenine monophosphate response element binding protein. Moving to next step. Cyclic adenine monophosphate response element binding protein seems to stimulate the activity of a target gene through its intrinsic histone acetyltransferase activity. The histone acetyltransferase activity is responsible for the acetylation of lysine residues in the N-termini of histones. 8 histone proteins can come together to make up something called a nucleosome, making the DNA template more accessible to the transcriptional machinery. So, this was a normal pathway. Now, let's understand the etiology. that the deletion creates a haploinsufficiency, which leads to this syndrome. Mutation in cyclic adenine monophosphate response element binding protein affects the further steps of this pathways and acetylation activity of histone acetyltransferase, and affects the final step, moving towards features: microcephaly, broad thumbs, and great toes flattened at ends and sometimes bifid or overlapping, widely separate from other toes, wide-set eyes, large convex nose, small mouth with high palate, teeth crowded. Moving towards management: correction of eye defects and undescended testes, surgery on feet, attention to obesity and dental issues, speech therapy and instruction in self-help skills. Last is Alagille syndrome, type 1. Frequency of Alagille syndrome, type 1, is 1 out of 100,000 live births. Genetics: Alagille syndrome, type 1. It is del 20, p12. It is autosomal dominant inheritance. 7% of patients have deletion of JAG1. JAG1 encodes a ligand critical to the Notch gene signaling cascade, important in the developmental regulation of bile duct formation. Deficiency of the JAG1 protein leads to a paucity of bile ducts and cholestatic liver disease underlying cardiac disease, CNS vasculopathy, renal disease. Let's understand Notch gene signaling cascade. This is signaling cell, and this is receiving cell, having Notch receptor. JAG1 gene provides instructions for making a protein called JAG1, which is involved in an important pathway by which cells can signal to each other. The JAG1 protein is inserted into the membranes of certain cells. JAG1 expressed on the surface of the signaling cell undergoes ubiquitination and binds to a Notch receptor expressed on the surface of the receiving cell. The core binding domains are JAG1 and Notch. Notch receptor subsequently undergoes proteolytic cleavage induced by an endocytic pulling force of JAG1. The full-length Notch receptor undergoes initial cleavage, which is S1 cleavage, and post-translational modification or glycosylation in the Golgi, from where it is trafficked to the plasma membrane as a transmembrane heterodimeric protein. In the Notch signaling pathway, when a Notch receptor interacts with a Notch ligand in an adjacent signaling cell, it undergoes proteolytic cleavage by disintegrin and metalloprotease domain 10, causing to S2 cleavage, and the gamma secretase complex, causing S3 cleavage, which leads to the release of the Notch intracellular domain. Notch intracellular domain is finally released and enters the nucleus and binds to the DNA binding protein, CSL, which recruits mastermind-like protein to activate the transcription of Notch target genes. So, JAG1 encodes a ligand critical to the Notch gene signaling cascade, important in the developmental regulation of bile duct formation. Deficiency of the JAG1 protein leads to a paucity of bile ducts and cholestatic liver disease underlying cardiac disease, CNS vasculopathy, renal disease. Let's discuss features of Alagille syndrome, type 1. Broad forehead, prominent chin, and elongated nose with bulbous tip. This was all. Thank you for learning. Join us again.
Watch our medical videos anytime and anywhere. Download sqadia.com app now.